we're going to do questions now, and then we have a little announcement, if you will, presentation from Jonathan. But questions now for Bill.
So I have a couple of comments, actually. Number one, we need to accept also that the reason why we may not have success is that we need better drug, right? The second thing is that, I mean, is a fact that along the line that you actually very clearly, I mean, shown, and I totally agree with, something that would be easy to look at and is not always looked at is the natural history of these diseases, right? You actually briefly mentioned this, but I mean, something that is never harmonized when you do studies in the advanced setting is the history of the patients. So they may have recur 1 year down the line, 3 years down the line, 6 years down the line, with one single nodule, multiple nodules, multiple sites. They are different patients, even if they have the same disease, and they may have also the same biomarkers. But still, I mean, what you are looking at is a different patient. And I think this is one other reason why many of the studies, large studies, that do not, I mean, that include all the patients that you see, all the cameras in your clinic, eventually miss the chance to see activities that may be there in subset of patients.
Yeah, I agree. I mean, it's all part of the process, right? Yes, we need better drugs, but how do we really use the science to get better drugs? And when we get better drugs, are we confident that we're going to study them correctly? Or are we going to follow the same process? But we have good drugs. I mean, I showed data with that, epithelioid sarcoma, PComas. We have good drugs, but we need the data to understand how to use them correctly. And also, how do we use those drugs to study the next generation of drugs? We're not doing that, right? So we also need to do that. And the next part of what you said was really important. How do we design clinical trials better when we can actually incorporate what we know about the patients and their disease into the clinical trial? And it's why I harp on the olaratumab. You know, this is a drug that received FDA accelerated approval, was being used for years in the United States. Every patient in the world wanted that drug. That's why we had a trial that accrued 600 patients in 10 months. But I can guarantee you those patients that we put on that study that rapidly accrued in 10 months were not the same patients that get placed on, you know, a randomized phase 2 study that took, you know, 4 or 5 years to accrue at, you know, 7 centers, right? So all of that is involved in it. But it's not as easy as saying we just don't have good drugs. We have to look at our processes too and say, how do we do better? And I think there are a lot of examples of that. We know rapamycin is a good drug. We know that one of the reasons we want to collect all of these data in rapamycin and EHE is so we can go to the EMA and say, let's get the drug available for patients, right? Because it's a good drug. May not be our best, but we can learn from it, and that'll help us study the next generation. I'm worried about John's comment.
So we've done a lot of doxorubicin analog studies in frontline. We've done doxorubicin combinations in frontline for soft tissue sarcomas. We've even picked a homogeneous disease like GIST, and we can't beat imatinib in frontline. So my question, or I'd like to get your comment on, is why aren't we studying doxorubicin analogs or combinations in later line therapy, where patients are sicker, they have faster growing disease, and any signal is going to be amplified. I know there's a theoretical concern of cardiomyopathy, but in clinic, we use doxorubicin in later stage disease. If, for instance, if a patient had an anthracycline as adjuvant therapy, 6 cycles, we'll re-challenge them later. Mm-hmm. And they, you know, there's not this huge amount of cardiotoxicity. So why can't we look at later lines of therapy?
I agree, John. And I think there's 2 components to that I'll touch on. First of all, polyphosphomide, GETIS study, ANNOUNCE, evofosfamide, aldoxorubicin. I guarantee there's over 1,000 patients with leiomyosarcoma treated in the first and second line study with doxorubicin where the data is out there. Why aren't we gathering that data and truly understanding doxorubicin in those diseases? Same thing with liposarcoma. Do we truly understand that? So I think that's the first thing, because we have to historically understand how patients have done. I agree with you. I think we should be testing a lot of these analogs, especially if they're thought to be safer in subsequent lines of therapy. The goal is always to bring it up first line. But I think it then requires our community to also say we are comfortable doing that. Because a lot of these drug companies, I suppose, are really worried about toxicity. And they're really worried about saying, you know, what do we do in a patient that's already had 6 cycles of doxorubicin? So how do we as a community get together and say, you know what, maybe that's where we should be testing it, right? And this is how we'll monitor for cardiac toxicity. And this is our collective data of going past 6 cycles. Because I think, you know, they're going to have the constraints of what they have to worry about from a regulatory process. But I also think we need data in our community to say it's okay to do this. In fact, maybe this is what we would recommend, right? Because if we see the signal there, it's easier to bring it up. I think the problem is so many of our patients are heavily pretreated with doxorubicin to that point. But it's a great point. But again, it's one of the messages where we shouldn't be at the whim externally of how we do things. We should dictate, you know, where our community should go, right? And the patient's voice is important for that because ultimately they need to go through it. But I think it's a great point.
Hi, Dave Schulman, Dana-Farber. Bill, I love this talk. I was intrigued by the comments you made on biomarkers towards the end, and I think, you know, you could hypothesize that those will help us find some of these signals of efficacy. I also think in rare diseases it's a challenge as you sort of slice the pie thinner and thinner, and I was wondering if you could comment on sort of, you know, using biomarkers in trial designs in rare diseases.
Yeah, it's critical, and I think, as you said, you know, the difficult thing is a lot of these are preclinical and scientific, and then we just don't have enough clinical data to use them, and that's, you know, really what's happening, I think, with CDH18. We'd love to show that more in liposarcoma, but you use different drugs, different patients, right? We look at tissue expression. We don't do landscape analyses on tumors. You know, these are large tumors, right? We looked for years back in patients, and so it didn't matter when it was positive, but these tumors are dynamic and can change over time as well. You know, if you have a 30-centimeter liposarcoma and you biopsy 5 areas, you may find 5 different things, right? So it is really challenging, but I will say, as the technology emerges, right, as we're dealing with ctDNA and circulating tumor cells, and the science is emerging, we have to, figure out, are we really doing that in phase 1-2 studies where we're beginning to generate the hypothesis, right? And that's why I wanted to show the chondrosarcoma and the PComa, not so much because anything there is definitive, but it's exploratory. But then how do we take that to the next level? And I think that's the most challenging thing. But if we can coalesce a little bit in the community, if we can identify biomarkers in a disease, you know, then maybe we can do that clinically and be consistent on clinical trials. The ASPS story is wonderful to me, but we also had, you know, 5 different studies running in parallel. You know, should we also create like an IIT database where the correlative work and the outcomes data comes in there, and then maybe when the next iteration of trials come on, you have one of these working groups of interest in the disease and say, let's really go through all of the data we have and say, this is how we can design a larger study, and these are the biomarkers that we could incorporate in there. It has to be done with the labs that are doing the work so we can iterate on it. But to me, that's the challenge, right? Because it's how do we support, you know, a lot of these efforts together. And I think we're moving to that. It's just how do we organize it, right? So it's a great question because it brings on a huge layer. And I think about that with immunotherapy, the hundreds of patients that are going on trials throughout all of our sites, all being done in isolation, all doing RNA-seq, all doing pre and post biopsies, all doing multiplex IHCs. How do we bring it together? How many patients do we need with osteosarcoma on a immunotherapy where we can't collectively and collaboratively say, how did they do, what did the tissue show, right? And I think that's where we also need to start. I think it's a win-win for all of the investigators, the patients, you know, in understanding drug development, right? So it's challenging, and, you know, maybe we could take some small steps, but adding the correlative work to this is paramount.
Well, congratulations on a wonderful talk. Kumar Sankhala from Los Angeles. So having a background in oncology drug development and sarcoma,
I see that our group has been lacking in two things. Number one, data extraction, and number two, not learning from other tumor types, that how the drug development is going in that sectors. Data extraction, Google knows that what flavor of coffee I drank this morning, but I don't know what is your second line and third line treatment for PEComa patient, and that's like for all tumor types. So I'm glad that you mentioned about that. The second thing is there's a lot going on, there's a lot of science going on, clinical trials going on in other tumor types. And in sarcoma community, we don't learn and we don't interact with those areas. So any comments about that?
Yeah, I think it's complex, right? I agree with you. I mean, I think the data is there, and companies now can extract huge volumes of data. And, you know, I've seen them run simulations in pontine gliomas, you know, with a drug, and you can totally see outcomes, right? It's really remarkable. And I'm talking about 11,000 records within 30 minutes the data is pulled, right? So then it's the community to say, how is it right? So the opportunity to do that is there. The way we incorporate that in a community is the hard thing. How do we start? How do we really move it through? The science is a tougher thing because the depth and the breadth of science that we have in our communities is, you know, really remarkable. And I think that's been one of the major advancements we've had in the last, you know, 10 years. It wasn't the purpose of this talk, and we do see, you know, people getting together and really talking about the science. But is that incorporated in the clinical trial designs? Right? Is that incorporated into our clinical efforts? And that's where I'll say, well, maybe we're missing the mark, right? Because we're often influenced by interest of a pharmaceutical company and a drug and a design, which is great because we want that interest, but maybe we don't always design the study correctly. And maybe we don't have the science, the correlative work at that table at that point, because there's a lot more that we can do. And I think we have to think about that better. I do think we have to learn what's going out there. I also think all of the work we saw today can influence other diseases as well. The cell cycle work I showed you, you know, is not just in liposarcoma, that was the model, but it's holding true in breast cancer, lung cancer, prostate cancer, gliomas, right? So there are that cross-pollination. So that becomes important, but difficult. But this is SARC, right? That's, you know, a call to action. They have the collective community sitting here. So how can we organize that with an organization like SARC? I don't know. Hey, Lee.
Lee Cranberry, University of Washington. Nice talk, Bill. I enjoyed you summarizing a lot of points in my career that I remember. What I think of this is, you know, we're going from an era where one size fits all, doxorubicin for everything, or one-size-fits-all trials, and ultimately going to be in an era, hopefully, where, you know, we know the biology of everyone and we can declare victory and go home. But we're in sort of this transitional period, and there's been some really exciting trials that you mentioned, but right now they're affecting maybe 5% of my practice. I mean, in terms of off-trial treatment, the majority of patients are the treatment I prescribe is still being directed on the basis of, I'll call them one-size-fits-all trials. And when I look at the ivofosfamide and olartumab trials, they were based on what was hopefully a very broad biological finding, but effectively they were histology agnostic. What's the role for those types of trials? What's our threshold, if you will, for doing those types of trials in this
I laughed at your bad pun, right?
Yeah.
No, but you're hitting the nail right on the head, and you know, none of this is to be prescriptive, but to get us to think about it, right? And I think you would be a perfect person to teach the next SARC seminar on these studies and where they went wrong, right? But I think when I think about sarcomas, it really fits into three categories. There are those with really specific genomic abnormalities that chemo plays no role, right? You know, your NTRK potentially, GIST we heard about today. Then there's, you know, those diseases that we just can't, you know, target what's genomically there. I mean, leiomyosarcoma, UPS, right? And then there are diseases that are in between that have known genetic alterations, but yet chemotherapy remains a hallmark of treatment, right? And those are diseases like liposarcoma. You know, we spoke about epithelioid sarcoma. So we need to start thinking about this, because I think chemo will always remain a hallmark, but then we also have to think about patient selection, we also have to think about sequencing, we also have to think about combinations, right? And how do we begin to do that? But I agree with you, chemo's not gonna go away, it really does work. How can we combine things? But it's why, I struggle with angiosarcoma, because chemo works for that disease. I had someone ask me the other day, it's a head and neck angiosarcoma with a low TMB. Do I start with immunotherapy or chemo? What do I do? Right? We don't know, right? So it's a hard discussion, but how do we begin to define that? I don't have the answer for it, but your thinking is right on, right? These are the questions we need to ask and begin to at least chip away at some of these other diseases. Same question that John brought up about, why are we using this first line? Maybe it should be second line, but maybe it's different for epithelioid sarcoma versus, what we would do in liposarcoma. So I appreciate it.
Thank you, Bill.
So Bill, one last question. As I look at all of these stories that you talked about, the one thing that fits a little bit into what Alex brought up earlier, like maybe we don't have good enough drugs if what we are looking for is a cure, is a complete response. And how many of I'm sure we're missing some of those specific pairings of a disease and a drug, or a specific, marker of a disease and a drug that are complete responses. But what I notice, and I, one type you haven't brought up at all, but is a pretty common type, is synovial sarcoma. Obviously, I spend a lot of my time thinking about that. We haven't found anything that cures synovial sarcoma. It's one of those, diseases for which we use a lot of, poison chemotherapy. We try to blast what is a very quiet genome and probably turn it into a less quiet genome, probably make our worst enemies for those that are almost always ultimately resistant to that, doxorubicin or ifos treatment. How can we look for a different benchmark? We're not looking for something that was stable over a long time, even pexidartinib. It's like, as someone who takes care of patients with tenosynovial giant cell tumors, I have no idea how to use that. I mean, am I just gonna put patients, I don't put patients on it myself. I have my medical oncologist put patients on it forever.
Yeah.
Like, how do we stop? Because that doesn't cure that disease. That doesn't have a complete response. That has an excellent response while they're on the drug. But then how do we get rid of tumors? So are there ways that we could change what we're looking at as a benchmark? We're not looking for something that holds it stable or shrinks it a little bit. We're looking for something that gets rid of tumors.
Yeah.
And there are occasional cases of that, but not many.
Yeah, this is the discussion we need. When we think about tenosynovial giant cell tumor, it's just great we were lucky enough to get a drug, but now we sit with our patients all the time and say, do you want to stop? Do you not want to stop? What do we do? And we learn from that. I think, when we think about synovial sarcoma, again, we should be defining what to do and how the studies should be and what we're looking for, and it may be different, with a first-in-class BRD9 degrader versus what we see with TCRs, or those extreme responders to pazopanib, but we have active drugs, ifosfamide and adriamycin, so are we setting the benchmark against them? Versus, do we have to think about the context of the patient, line of therapy, the inhibitor we're using, and then how as we as a community get together in some of the synovial working groups and saying, here's the white paper, here's the standard of care, we can take this to the regulators and say, no, we're not looking at RECIST responses or complete response. What we feel is important is X, and then hold that. Because so many of these trials, the outcomes were not dictated by the community, and I hate to say it, as much as we spend time talking with the regulators about what we need to do in ultra-rare cancers, we just can't prove or validate those systems. It was like the TVS and TGCT, we still had to use response. Not that the TVS was better, but it was different, right? So how do we take that upon us to begin to think about the outcomes for patients? What empiric data do we have? What is the line of setting that we're treating in, the inhibitor that we use, and begin to define that? How do we use your science, right, with BRD9 degraders, right, the BAF complex, and say this is how it should inform what we see on those studies, right? Because otherwise we'll either see a signal that will overinterpret or see a signal that will underinterpret, and all of that affects the drug. How do we build biomarkers early on into those studies so we can see who's responding or not? So this is the complex questions that you and Lee and everyone brought up to say, what is our responsibility as a community? Is everything in isolation by who contacts us and what we're doing and what's moving on and our interests? Or can we begin to coalesce and then dictate outwards, right? I think that's where these discussions should go.
Thank you very much, Bill. We are going to move next with a perfect segue to how we can be strategic